Welcome to Science Saturday, EHE Research and Momentum. Thank you for joining us today. For those that don't know, my name is Maggie Cameron. I am the Director of Development and Communications for the EHE Foundation, and I also have EHE. This is a very special day. It's actually Rare Disease Day today, which is a global day of awareness for the over 300 million people living with rare diseases. NORD spearheaded this movement to advocate for equitable access to diagnosis, treatment, care, and social opportunities. And here at the EHE Foundation, we believe that a great way to honor Rare Disease Day is to provide a thorough update on the state of EHE research. So we welcome you to keep your cameras on, unless there's a lot of movement going on around you, or if you need to step away, of course. And please keep your audio muted except when you're actively talking. The chat function is available to you, so feel free to use it to communicate with other attendees and put your questions in there as they come up. We will leave a good block of time at the end for Q&A. We appreciate that you might have questions about your specific disease, progression, or care. We cannot answer those and encourage you to take those questions to your care team. And along that same line, we want to remind all participants that we cannot provide medical advice, and the information provided here is for your information only. All right, let's get started. Your presenters today are the other 2/3 of the EHE Foundation staff. We have Executive Director Denise Robinson with us, and the newest member of the team, Director of Research and Engagement, Dr. Christy Aristian.
Okay. Maggie, thanks so much. Welcome to Science Saturday, everybody. I see familiar names. I can't see your faces right now. I'll see your faces after we get through with the slide sharing. But I want to say welcome and thank you for making time to be here today. I'm Denise Robinson. For those of you who don't know me, I am the Executive Director of the EHE Foundation. And as Maggie said, I'm going to be joined in today's conversation by Christy Aristian, who's the Director of Research and Engagement here at the Foundation. For those of you who don't know the Foundation, we are a nonprofit advocacy organization. We are dedicated to research and supporting patients and finding better treatments and a cure for EHE. And I want to first just start out the day by saying thank you. I want to thank people who have taken part in research, people who are taking part in research today, and that looks like many different ways. I mean, there are so many ways to take part in research. And I want to thank the care partners, clinicians who guide patients in research and help patients get through research, and then our scientists who are in the lab, and you don't often get to see them. They kind of are the unsung heroes in research, and we want to thank everybody. Especially, I want to thank our donors, because without donors and supporters, we couldn't do the work that we are going to talk about today that's going on all over the world. So we know that this is not a community that you wanted to join. It is a great community of support, and by your presence here today and in the research space, you're helping make progress. So thank you. As Maggie mentioned, I want you to put questions along the way in the chat function on your screen. We're going to leave plenty of time for Q&A throughout the day. Today's conversation is a journey, really. We want to take you through what we know. We want to share what we've learned, what has been accomplished. There's so much that's going on. I really feel, I want to say to you, this really just scratches the surface. I could talk to you for days and days on end. And there's so much literature out there now, much more than there was when this foundation started. We're going to talk to you about research directions, resources, and tools that we have, some that we still need. We'll talk about clinical trials and other studies and ways that we're going to learn about EHE and about treatments in order to get better treatments. And we'll talk about future perspectives. So again, just put your questions in the chat. We'll get to those later on. And right now, I'm going to turn it over to Christy.
Thank you, Denise. Good morning, everyone. I'm Christy. I'm so excited to get to meet you all today. I'm going to start with what we know about epithelioid hemangioendothelioma, or EHE. We can learn a lot about the basics of EHE from its name. On the right, you'll see this little diagram here. Epithelioid refers to the kind of cells involved in this cancer. The prefixes there, heme and angio, tell us blood and blood vessels are important, and the endothelial word refers to the specific cells that line the interior walls of those vessels. Last but not least, that suffix, oma or eoma, tells us that this is an abnormal growth or tumor. So together, the name of the disease says that this is an abnormal growth of the cells that line the inside of blood vessels. But there's obviously a lot more to EHE than just that. It's an ultra-rare vascular sarcoma, and by ultra-rare, we mean it occurs in less than 1 in 1 million people. To diagnose EHE, there are important features that we look for. The first is the presence of one of 2 fusion genes, or fusion proteins, that really are the defining features of EHE. And those 2 are WWTR1, sometimes referred to as TAZ, CAMPTA1, or less commonly YAP-TFE3. If you've been diagnosed with EHE, you may or may not have seen those on your pathology report, but what you almost certainly did see is immunohistochemistry, or IHC, for some key proteins that are characteristic of the tumor. Those are CD31, CD34, and/or ERG. Like the blood vessels it's named for, EHE can grow in almost any tissue, but most often is diagnosed in the liver, lungs, and bones. A unique feature of EHE is that this is a heterogeneous disease that's very diverse in presentation as well as who can have EHE. It doesn't look or behave the same from person to person. The people affected by EHE are different and unique, and it can affect people of all ages, including the very old and the very young. Both men and women can have EHE, but it seems to affect slightly more women than men. And the disease can grow very slowly or be very aggressive. In fact, over 50% of people have metastatic disease at the time of their diagnosis. With both localized and metastatic disease, some people experience no symptoms at all, but if they do, those symptoms can be varied and similar to other diseases. This is a challenge that it especially presents itself during the diagnostic journey. EHE really is not just one disease. It's many different experiences of the disease by different people in different ways. And now I have a challenge for you. Think of 3 words to describe a home. I'll give you a few seconds to think of those 3 words. Okay, have your words? Now I'm going to ask you, are the words you chose representative of all of the houses you see here? Is there a kind of house shown here that you didn't think of? And now that you see the whole picture, would you choose different words or the same words that you previously chose to describe all the houses? This is the challenge of research in EHE. To guide research, there are broad, overarching questions that help us think about it. What are the common features that are true of all EHE? What are features that are true of most EHE? What are features that are true of some EHE? How do we identify and describe those groups of people? And then once we've done that, what interventions are best and most effective? I encourage you to remember this exercise as we talk about some of the data and what we've learned. These overarching questions can lead research in many different directions. So how do we get where we want to go? Well, we do that by investing in research in strategic areas to keep fueling that research pipeline. As we look around the globe here on your screen, we see different kinds of research that all must work together to get us where we want to go. As part of our discussion today, we'll touch on the work going on in each of these areas. I mentioned the basic biology of the WWTR1 and YAP1 fusions, and next I'll introduce some of the progress that's been made in developing resources and reagents, such as the cell cultures, PDX models, and genetically engineered mouse models. We'll share some of the important translational science being done by researchers studying repurposed drugs and identifying promising pathways making progress in drug development. We'll also go over some exciting opportunities in the form of clinical trials, testing new inhibitors, and new drug formulations. And finally, we'll talk about how real-world evidence in the form of registries and observational studies is fueling discovery and shining light on the next steps. And throughout all of this, we'll consider where there's opportunities that still exist to make progress exploring biomarkers of prognosis and treatment efficacy. Each of these areas of research is required to fuel progress, and progress in each of these areas is dependent on continued investment in the others.
So I want to jump in here. As Christy highlighted, there are a lot of areas that we've made progress, and there are so many areas where we still need progress in order to find better treatments and learn more about this disease. So here I just want to take a moment to shout out that we've invested as a foundation now almost $3 million in highly impactful research. I also want to say and acknowledge our advocacy partners globally, the EHE Rare Cancer Charity UK, the EHE Foundation Australia, EHE Italia, and the Canadian EHE group, who are also funding impactful programs in their regions. And I want to reiterate, everything that we're talking about today is possible because of donors, because of really passionate and talented researchers and participants who take place in research. And research programs take years, even decades, really, to yield actionable results. And Dr. Brian Rubin's lab at the Cleveland Clinic is a flagship for persistence in EHE research, really yielding important biology, forming meaningful and important collaborations with other labs like the Lamar Lab at Albany Medical. These are things that have taken years, and Dr. Rubin really has been studying this for over a decade, and his lab remains committed to finding treatments and are currently studying ways to treat aggressive EHE. So we look forward to learning from all of these centers. The work that's going on here really is going to keep going on for years and years, and through those years it will continue to yield important data that will guide us in our knowledge of EHE. So I think that through partnership, through collaboration, by showing up and staying engaged in the clinical care community and in the research community, we have already made progress. Already, patients benefit by the learning, so clinicians can guide better in those care journeys. Researchers have tools and methods that they can lean on with collaborators and borrow from, and we want to make sure that that work continues. So with your support, we're able to do that.
And as Denise said, this work really has generated a lot of progress. Those years of investment around the globe led to the development of several important models of EHE, and these models are some of the most tangible results from our collective and collaborative research efforts. Dr. Rubin's lab at the Cleveland Clinic has developed a genetically engineered mouse model of TAS-CAMTA1 fusion-driven EHE. In Milan, Dr. Pasquale has developed a patient-derived xenograft, a model of human cells grown in mice. Dr. Tanas's lab at the University of Iowa has developed cell cultures from samples donated to the EHE Biobank. All of this made possible by individual people donating their tissue. And other groups are now able to work with cell lines that have been grown out in dishes from all of these models. But there's still work to be done in this space. We need different models to represent the different forms of EHE. Each of these models have different uses, different strengths, and different weaknesses when answering research questions. We must continue to make models accessible to the research community to facilitate collaborative science, and the most impactful models are made possible only if real people with EHE are willing to contribute their tissue. To understand all of the different faces of EHE, we need diversity in models and diversity in the people who contribute to them. This is a diagram from a recent publication from Dr. Pasquale and colleagues in Milan, and what you see depicted here is how information from the lived experiences of EHE patients can be used to develop more accurate and representative models of EHE, from which we gain valuable scientific knowledge that can be applied to the development of clinical trials, which then offer new opportunities for care for patients. This process really starts and ends with patients. EHE model development is challenging. It requires different people with different presentations of EHE to contribute. Denise, did you want to talk a little bit about the table here from our own EHE Biobank?
Yes, I do. Thanks, Christy. Many of you know we started the biobank at the EHE Foundation a number of years ago, and I just want to highlight. It is very challenging to get samples for development of models, and the EHE Biobank is not the only way to do that, but this is why we we developed this biobank to solve this problem of model development. You can see, if you can read the tiny numbers, only just over 10% of what we collect is actually usable for the development of models. And it takes a lot of samples actually to develop a model, as it turns out. So we're really focused on this and making sure that we are driving this progress as best we can. It's just a numbers game. It's hard to say that out loud sometimes, because it does start with patients to bring this progress back to patients, but it takes a lot of numbers, and we all know that this is a fairly small community. I want to jump over and talk just a little bit about the biobank. First, I want to say a huge thank you to the people who have joined the biobank. This is a picture of Maggie, and Maggie self-identified. She raised her hand and wanted to celebrate this and invite other people who have EHE to join her in donating their tissue. If you've donated tissue to the EHE Biobank, you are a game changer, and you can be a game changer. Everybody can want to do this. Not everyone has surgery, so there are limitations on what people can and can't do. But when we initiated the biobank, we knew that there was a big problem that needed to be solved, and we didn't realize how hard this would be, but it is good work to be done, and we're glad that we're doing it. This project really exists to make sure that labs can get tissue for model development, but for other analysis that need to be done. And you might think that your major hospital, if you have surgery or a transplant at your hospital, you might think that tissue that is taken from your body goes directly into EHE research. But more often than not, that's not what happens. It may go into general cancer research, but it probably does not go into specific EHE research. And we need every surgery, every specimen that we can get to go into EHE research to help find a cure and learn more about different people. As Christy mentioned, the heterogeneity of the disease. We have to be able to learn about different presentations of the disease. And the only way that we can do that is to study those cells and make models of those people. So I urge you to scan the QR code. It is not a commitment. You are joining as a participant, but there's no obligation until you have something scheduled, and then we can work with your care team with your consent. We're glad to answer separately. If you scan, you can contact us and we can answer any questions that you have about the biobank.
Denise, may I just make a clarification? Sorry, it's Fiona. Just that all of this wonderful information you're giving is specific to US patients. And so if this is an international group, I don't know where everybody's coming from. It's important to note that US patients, please, please, please support this initiative. Otherwise, check with your local group. Canada does have a biobank out of Mount Sinai Hospital in Toronto. So I just wanted to clarify that, because this is such wonderful information, but I know that not everybody can contribute.
Thank you, Fiona. That is a good call out. We can take specimens from other countries, but it is more impactful and probably easier if you are in Canada, contact the Mount Sinai team. If you're in the UK, you can contact the Royal Marsden. There's a biobank there, and there's a biobank in Australia. You can contact the EHE Australia Foundation about those. They all operate differently, so in research, everything is unique. Each project is special. But you can get in touch with us at the EHE Biobank. And if you're not in this country, or there is somewhere that is more appropriate for you to donate tissue, we can put you directly in touch with those people too. So thank you, Fiona, for that call out. I think we are back to Christy for the next portion here.
We are, yes. So models aren't the only way that we're asking and answering questions about EHE. There's many, many pathways to progress. There's been a lot of research interest around the hippo-TEAD pathway. Many of you have heard about this before, probably, and that work is gaining a lot of momentum as well. When it comes to cell and organ growth, hippo signaling is sort of like a brake pedal, and TEADs are the accelerator. When Hippo is on and YAP and TAZ stay away from TEADs and genes that cause growth are turned off. When Hippo is off, YAP and TAZ connect with the TEAD proteins and genes that cause growth are turned on. Both the accelerator and the brake are important to drive the car safely and correctly, and healthy cells understand when to use the accelerator and when to use the brake. In cells with EHE fusions, the fusion proteins push on the accelerator continually and never use the brake, causing the car to speed off out of control. The goal of TEAD inhibitors is to try to stop that out of control car by pulling back on the TEAD accelerators. The trick in developing these drugs is finding the right balance so that healthy cells that have the same accelerator and brakes as EHE cells aren't damaged too. We're encouraged by the TEAD inhibitors that are in early stage development, and Denise is going to share a bit more about that clinical work in just a little while. The EHE fusion proteins are the most common and arguably most important mutations that we know about right now, but other lanes of research have shown that they are not the only mutations that occur in EHE tumors, and this may be meaningful for people with EHE. Some of you may have also heard about CDKN2A as another important mutation in EHE. In a 2019 study led by Dr. Chen at Ohio State University, they found that more than half of the patients in their study had secondary genomic mutations in addition to an EHE fusion, and the most common of those was the loss of this CDKN2A gene. Then in 2023, Dr. Neal and Dr. Seavey showed in 2 separate models of TAZ-CAMTA1 driven EHE that loss of CDKN2A combined with the fusion allowed for uncontrolled cell growth and produced more aggressive EHE. Loss of CDKN2A as a gene is a more common mutation in many cancers, and together these studies have suggested that in EHE, CDKN2A loss is associated with more aggressive disease. However, we do not have a standard clinical diagnostic tool that we can use to detect this gene mutation in patients. One option is IHC for a protein called P16, which is one of multiple protein products of the CDKN2A gene. Another option is genomic sequencing, which, although a more complex and expensive test, is highly specific and can reveal information about genomic mutations such as CDKN2A. Sequencing can be a very important tool as part of the EHE diagnosis and prognosis process.
So another area that we have learned a lot in the past, 5, 6, 7 years now is this growing case for sirolimus in EHE, an mTOR inhibitor. So sirolimus is an mTOR inhibitor. It's been studied in many diseases and has been used for years to prevent organ transplant rejection and to treat rare genetic conditions. In 2019, sirolimus emerged as an effective treatment for some people with EHE. This was reported in a small retrospective case series from the Italian Sarcoma Network. Their evidence of use of sirolimus in EHE grew, and that research now has grown significantly at the INT in Milan under the leadership of Dr. Silvia Stacchiati. Additionally, Dr. Nadia Zafroni and Dr. Sandro Pasquale have revealed preclinical data demonstrating the superiority of sirolimus to doxorubicin in a PDX model. You can see that in the center graphic here. And this is all important because this growing body of work is revealing biomarkers. It's discovery of other biology of the disease. Dr. Lamar's lab is represented in this research. So you can see through more data supporting the investigation of sirolimus in EHE, and this grows, and we expect over the next few years, where there's a large observational study to continue to see data readout on sirolimus for EHE. Importantly, you'll see at the bottom right, there's a new study that's being started, and that is to investigate nab-sirolimus in EHE. So this is a new formulation of sirolimus that may be more targeted and have less toxicity or side effects for patients. So we'll talk a little bit more about that study in a little bit, but I want to make sure that you can see, because this is a common question that we get when we are talking with patients, and we know that this is a question even for doctors of what agents are more effective in EHE. So just know that there is a growing case for sirolimus, and this case will continue to build as we get more data.
Denise, I also just wanted to draw attention to that graph you have shown there. It's a great example of how researchers are using patient donated samples to ask and answer research questions. That PDX model was generated by growing a person's EHE cells in mice, and now we're able to see how those particular cells responded differently to the two drugs that were studied. It just goes to show how important it is to be able to study different kinds of EHE in the same way.
Mm-hmm. Good point. So what have we learned about sirolimus so far in EHE? So we know that there's preclinical evidence to support mTOR as a therapeutic target. That's well supported. We know that there's preclinical evidence, as we just saw in that graph, that sirolimus was superior to doxorubicin in a model system. This is a drug that we know that people are treated with doxorubicin, so that could be important data moving forward for to translate into clinical care. We do know if you go through all of those studies, not all people benefit from sirolimus equally. We know that some presentations of sirolimus, some presentations of EHE aren't as well controlled with sirolimus as others. So the context of each study and of each patient is important. We know from those data that sirolimus might offer prolonged disease stabilization, which is important. There are people that are, if you're diagnosed and you have an indolent disease or slowly growing disease, and people worry about being on a medication for a long time, will it be effective? Can I do that? So there's growing evidence of that, and we need more years of data to be able to support that. There is data that sirolimus has demonstrated in a very limited data set to stabilize progressive disease. So people who have that more advanced disease, there is evidence of that. So that's very important for people. We know that patient access is important, and this is an area of great need. We did a survey back in 2024, and you can see in the graph on the far right, it's actually the bars that are in the left, that access to sirolimus is very important to people. So more data is still needed to expand the labeling, to change the guidelines, and to ensure that patients get access. So a large challenge is just making sure that we have good data to support this, so that we have good conversations and presentations to regulatory bodies, but also that clinicians have data that they can use in decision of care.
So the sirolimus story that Denise just spoke to is a great example of how existing drugs designed for other purposes might be repurposed and applied as part of the care of people with EHE. Here in this slide, I'm showing you two other examples where investigations in model systems have led to the identification of classes of drugs that may have some utility. Dr. John Lamar's lab at Albany Medical College has done work evaluating preclinical models of EHE and looking for compounds that seem to impact EHE cells. That work found that statins may be a therapeutic option for some kinds of EHE, but it also raised some important questions. Can combinations of statins with other drugs treat EHE? Can statins, which have been well studied and shown to be safe for many years, be used to control indolent EHE and prevent it from becoming aggressive? Do statins improve outcomes in EHE patients? These are all research questions we're still trying to answer. Similarly, Dr. Pabati at Cleveland Clinic has done work that identified a class of medications called CDK9 inhibitors. Dr. Pabati's research in cells and in mice has shown that these inhibitors may present an opportunity for EHE patients, and further work in this area is being supported by the NCI and SFA along with the EHE Foundation. This research illustrates how both new and existing compounds can be investigated for utility in different kinds of EHE.
Okay, so let's talk about clinical trials. We know that this is a question and a topic that's on many minds. And let's talk about other ways that we're learning about treatments in EHE as well. So there are new therapies in development. This is an exciting time for many of you. If you've been in this community for several years, I hope that you are as excited about this as I am. Today, there are more trials than ever for people with EHE, and for people with advanced disease, who are considering systemic therapy, I strongly encourage you to talk with your doctor and include clinical trials in your care decisions and your treatment choices. Clinical trials should be considered as a treatment option for people who have limited treatment options. So the first thing we're going to talk about are TEAD inhibitors. So this is a new and potentially important class of drug that have been in clinical stage development now for 3 to 4 years. These are early phase studies. Not only could TEAD inhibitors help people with EHE, but they hold a great potential to benefit people with other cancers and other diseases too. So this is an important lane of development. So far, we've seen 2 TEAD inhibitor trials open. We saw them accrue and enroll patients. People participated from this community, and we saw those close. The IKENA Oncology program with IK930 and the Novartis IAG933 trial. We were glad to see those products come into development. We are very sorry that they've closed, and we are really grateful to the patients and the research teams who participated in those projects. Today, we're excited still. We have TEADs in development, and we are, moving on with those. These are all slightly different compounds. So TEAD inhibitors, I'm not going to go into these mechanisms, but they're all different. These studies, everything that you're seeing here is being studied for people with advanced EHE. So these are for people who would need systemic therapy or who potentially could benefit from systemic therapy. The first one that I want to highlight here is the TEADS trial. It's the TEADES trial of a TEAD inhibitor. It is actively enrolling in EHE patients in the US and Europe. I believe that Orion is working to have other countries join this study, but we're really encouraged by very small data that Orion shared at a poster at a conference that we went to in November. And again, very encouraged, and we believe that this program is invested and poised to learn more about EHE patients. So there are new locations that are opening up. I urge you to, you can go to our webpage, the clinical trials webpage on our website at fightehe.org, and then the clinical trials number, that NCT number that is shown on each of these, you can go to that at clinicaltrials.gov. It'll tell you a little more about the study design, it'll tell you a little bit about the inclusion-exclusion criteria. I will caution you that not all of those pages are up to date with the study site locations. So if you'd like to learn more about some of those locations, contact us and we'll share that with you as best we can. The second study that's listed here is the Vivace study. I want to highlight this because, again, we know through patients telling us that they've enrolled in this study. That study is not currently enrolling EHE patients now. It was open in the US and Australia, but they're not enrolling new patients at this time. Their current focus is on mesothelioma. We are pressing them hard to make sure that they are still focused on EHE because we believe that this is a need and we want this to continue in development. So stay tuned for news from us as much as we can get. And we'll share that back with you. But we are encouraged, again, I mean, they're studying this drug for mesothelioma as well. And so development in another disease can still help this patient community. The next study that I want to talk about is the SpringWorks Therapeutics study. That's the third one down. So this is a newer, they're earlier in their development program. They are still in dose finding when we last spoke with them, and we're not focused specifically on EHE patients to include in the study. However, at this time, if you meet the criteria and you wanted to join this study, someone could join this study as long as they have slots available at the study site location. But we expect that they will grow in their development program, and we'll keep you updated on that as they do. The last TEAD study that's listed here is the Bridge Gene Biosciences TEAD inhibitor. This is the one that we know the least amount about. We know it's open, we know it's enrolling, but we don't have a lot of interactions with them. They haven't said a lot about their study, and we're still working to find out more about that. So stay tuned about that, but we are encouraged. So of those 4 TEAD inhibitors, the Orion one has the most activity right now and the most availability for people to be able to join at multiple sites around the world. So that's really good news. Then I want to jump down to a couple of other studies that are enrolling now for EHE patients. So again, this is really exciting. We've never had a time when this many studies we could point to that specifically included EHE as target populations. So the SARC-046 study is newly initiated. It just opened last week at Dana-Farber. It is going to open at 5 total centers right now in the United States. So this is a phase 2 study that's going to study nab-sirolimus. So remember, we talked about the case for sirolimus a couple of minutes ago in patients with EHE. So we're really encouraged about this. So nab-sirolimus is a different formulation. This is going to be sorry about that. This is going to be an albumin-bound formulation of sirolimus. It is an IV administered drug. So if you take sirolimus today, you're taking that orally. This is an IV administration, and the idea is that the drug targets the tumor but more than the normal tissue, so you don't have that systemic effect, and it would be less toxic. So ideally, more effective on controlling EHE tumors and less toxic. So that means less side effects for patients. So we look forward to this study opening up at more locations so that we can learn more about this drug. This could offer another benefit where people maybe don't benefit from sirolimus. Maybe they can't tolerate it. We don't know. It will take a couple of years to get this data in, but we'll keep you updated as slots become available and sites become active for that study. And then the last study on here that I want to highlight is an important one. This is out of Canada. This is out of Princess Margaret. So this is a phase 2 study of a PD-L1 inhibitor in patients with angiogenic sarcoma, including EHE. So it's a small study, but a PD-L1 inhibitor. This is an oral immunotherapy drug that is being tested in EHE. So this is the first, to my knowledge, focused study on this type of product in EHE. So we're really excited to learn more about this from Dr. Razik and his team there. This study is very early in its startup. It is accruing patients, again, only for people in Canada, and it's available at Princess Margaret. So if you'd like to learn more about that, again, you can go to the NCT number for all of these studies and read more about them and get in touch with us, and we can share more about that with you. I will point out really quickly, so the SARC-046 study I mentioned, we are planning to go to Dana-Farber in April. We'll have information about that in probably in the next week. We're still waiting for confirmation on some of the details from Dana-Farber, but we would love to see you there. Look on our news channel and on our social to see what we say about that in the coming week. So I want to talk a little bit more about systemic agents. So everything that we just talked about were clinical trials for novel treatments for EHE. So these are things that haven't been tested. They're not currently approved for use, and in most cases not approved at all for treatment of patients. So we still need to understand, though, how existing treatments work in different presentations of EHE. So right now, we all know that people are prescribed different things. There are different opinions on what to prescribe for EHE. So we need to learn more about these drugs as well, because they will continue to be used to some extent. And if they shouldn't be used, we need to learn that as well. To date, there are only 3 clinical trials that have been completed in EHE, 2 that had a reasonable number of patients, and then 1 recently with a very small subset of patients in eribulin. It's important to note that none of these studies met their primary endpoints. That means that they didn't meet the control rate sufficient enough to continue that area of investigation. Although all of them had very good learning and point toward reasons to study those drugs further. Some patients benefited from those drugs, so that really points to what we always say is we need to study that further. The table that I'm showing you here, Dr. Sylvia Stacchiati shared this table previously. It's been used in our conversations with EMA. It was used illustrating the case, the growing case for sirolimus in EHE. You can see the bottom row of data talks about sirolimus in 38 patients in EHE. And the important thing to point out here is the sirolimus data is retrospective data. And what we learned through discussions with regulatory agencies, the EMA and the FDA, is this is not the type of data that they are interested in taking. So if they're considering how they would approve even an existing drug, a drug that's FDA approved for EHE, or a novel drug, they want to see prospective data. So we need different study designs. But the thing
Sorry,
to know,
Denise, can I just ask for clarification? I'm not so for the broader group. What does ORR, MPFS, and PFR mean as we're reading this chart, please?
so yes, thank you, Fiona. And my point was not to go into all of those different data specifically, but you're looking at response rates. So the middle, that ORR, you're looking at response rates, and I won't go into the specific definitions of how these are calculated. The MPFS is median progression-free survival, and then you have progression-free response at the end. So I'm not going to go into how all of these are calculated, because the important thing to note here, and really the reason behind this slide, is to say that all of these data are captured under different conditions. So you have data on each agent that was captured under different study designs. So some of it is retrospective, some of it is prospective data. There were very highly variable criteria that were used to select the patients for these studies, not consistent across each thing. And so when you have variability in the data in the patient populations that were treated and the patient populations that were described in these data, that leaves a lot of interpretation of the data, and it doesn't answer really the questions that we need. And really where this leaves a burden is on patients and on clinicians, I would point out. So as we talk with patients, as clinicians are making decisions, as you are presented with an option, we often get a call that says, what is the best treatment for EHE? And the answer is it's hard to say. And it's hard to say because these data are so different that you can't definitively say that one thing is significantly superior to another. Some of the data if you look at the numbers, some of the data look close, and then some of the data look very different. But these are all different data sets. So what I want to point to, and what I'll continue to talk to Fiona, is that we're really working to get high quality data. And that means studied data so that we can advance the treatments. And I really caution you in any interpretation of this data. It's kind of my red box warning on this slide. But it is important to know that this data has been collected, because data does point us in new directions, and it will help us ask other questions and then figure out how we can better answer those questions going forward. So I want to talk a minute about observational studies and registries. So this is how we're going to find better data. So we need this real world data because, there might not be a study that comes up that wants to study a particular agent. I'll point to doxorubicin because it was previously mentioned. Trametinib is a drug that has previously been studied. But we may not have opportunities to study those in focused phase 2 studies again for a little while, but through real world evidence and data, we can learn about how treatments work in EHE. And I'm going to just put a note that active surveillance is also it's an approach to care, and we believe that that should get as much study. It needs to get as much study as any intervention, because that's how we learn what the disease does left alone. So I want to talk about this real world data that we're trying to get, and I can't tell you enough. It's hard to describe how much effort is going on into each of these projects. We at the foundation put a significant amount of emphasis on this because we believe this is one of the biggest things that needs to be undertaken in this community, and we're trying to drive new projects and expansion in these projects. And sometimes expansion is more locations or more participants in these projects. So one of the first ones here is the Eurocan registry. This is a registry that is only available in Europe. It's important to note that for patients, you can't raise your hand in Europe and say, oh, I want to be in that study. The doctor at the site where the study is open, it would invite you to be in this study. So this again is clinical data. So there's a form that the clinician fills out that answers specific questions about the patient, about their journey, about treatments, presentations, lab values, etc., to learn data about patients. The second one here, the POEM EHE observational study. So I mentioned this one earlier. It's in the case for sirolimus. This is where this really is right this second. This is a huge observational study that has multiple sub-studies. And aside from describing the disease, it also is going to help in uncovering biomarkers and building the case for sirolimus through those sub-studies. Again, like the study above it, patients can't raise their hand to join this study. As people are diagnosed at these locations, this study you will be invited to consent and join the study. It's currently open in Italy and the UK. It is planning to open in Canada, in the United States, in Taiwan. So we're working with researchers everywhere to try to get more patients in this study, because this is a really robust data set, has translational implications, long-term implications and benefit for care guidelines. This is a great body of work. This project is co-funded and supported through multiple advocacy organizations, and we'll keep that going forward. The EHE Global Patient Registry is I would say the most unique one on this page, and the one that I'm most passionate about, because this is our best opportunity to build the largest data set. Any person who's diagnosed with EHE or their care partner, their legal guardian, if you have access to the internet, you have an ability to raise your hand. And at a minimum, if you just say that you have EHE when you were diagnosed, have some representation of where your tumors are, where you are in your disease stage, that alone helps us understand the real impact of EHE around the world. So this is a really, it's patient donated data, so different than everything else that's represented here. This one is patient data, and we believe that this is important to learn more about this disease. And then the last one is the Excelsior study. This is a partnership that we have with XCures, who they're trying to extract data from medical records. So I've got USA as where this study is available. Technically, anyone can consent to join this study, but it is really hard to get records outside of the United States. It's a very heavy resource lift to do that. So we've focused on the US primarily, and I'll talk a little bit more about that. But what I want to point out here is that the good reason that we're putting effort into these types of studies is that these are data that are required for regulatory considerations and decision making. These are data that can be used no matter what regulatory bodies may decide. They can be used to change care guidelines, treatment guidelines, so they can be important absent of any regulatory decision making. And they're really essential for drug developers. So when we're talking about Orion, Vivace, SpringWorks, all of these companies need this natural history data data to understand the disease. When they come to the foundation, when we talk, every single drug development company that we talk with, they say, what natural history study data do you have? And that's the data that they're going to point to. So we can't encourage people enough to take part in these studies everywhere that you can. Dr. Rozic has a growing body of data in Canada. We'll look forward to sharing more about that. There was a recent publication on it. There are publications popping up all over the world, and where we can celebrate that data and help leverage that data for all of these interactions, we'll continue to do that. I just touched on the Global Patient Registry, and there's a QR code here. I encourage you, if you haven't yet joined the registry, scan this code. It's a super easy process. I'm not a patient in the registry, but I've taken these steps, and everyone has said this is super easy. This is just to describe people over time. Again, I say at a minimum, if you've been diagnosed, just join just to let us know that how many people have been diagnosed. Every bit of data helps us. It will describe treatments and outcomes over time. So we say this is a longitudinal database. That's over time, we're going to learn about you and how your disease changes. And it's not for any one person. It's really to look at multiple people. And then as we get more people, then we can start to do analysis to understand how it affects those people. We point to this data set as one way to identify patients for clinical trials. So today, that has already been important to identify people to take part in different types of research, clinical trials, and other types of studies that we support and collaborate on. So please make sure that if you can, we invite you to join. Here's a little snapshot of what we have in the registry today. It's hard to sit here. The registry's been open since 2023, I think. And you would think that we have more data, but it is a really rare disease. It's an ultra rare disease. And there are a number of people who don't have treatment. There are people who do have treatment, and all of that is different. So right now, we're really excited that we have 31 countries represented in the registry. That's amazing all by itself. We have 70% representatives by females, 30% males. That is not to draw any conclusion whatsoever that EHE is disproportionate in males to females to this percentage. This is basically women signing up for the registry more than men. So we would encourage more men to join our registry. Certainly, we need data from everyone. There are long-term studies on participation in this type of study, and it does show that women participate more than men. So we think that that's what this data is. We're not drawing any conclusions about who's affected by this disease. So far, 60% of people in the registry report that they've had a treatment or an intervention. 40% report that they have had no intervention. 17% are on systemic therapy. And again, way too early to do any analysis of that data, even to point to what are the more systemic agents used, because you very quickly get into ones, an N of 1 on use on any agent. We have 18 people that have reported they have liver transplants, and we really look forward to seeing that data grow, not only in the numbers of liver transplants, but in the years lived after that liver transplant. That's what we want to see grow. Right now, 43 years of age is the average age of the person. That's the average diagnosis age of people in our registry. So we have people that are much older, people that are younger, 43 years. 28 years is the longest. That's the most years of lived EHE that we have in the registry. And I'm going to put a note here. The 5.6 years of average years living with EHE, we believe through looking at our data that number is lower because we have more newly diagnosed people than long-lived people in the registry. So if you have lived with EHE for 10, 15 years, 8, 9 years, it doesn't matter if your disease has not changed. Even if you had a surgery 10 years ago and your doctor said you were cured of EHE, your data really can help us drive this number and learn more about how long people live with EHE. So again, I encourage you to join the registry if you have not, or consider that. You can learn more. Okay, that was a lot. I'm going to hand it back over to Christy for a second.
Thanks, Denise. You actually teed me up great because you just referred to the fact that we can kind of only learn from what's in our registry. Well, the same thing is true in AI and health AI and research. Just like real people, just like us looking at data, AI can only learn from the data that's available. And currently, the EHE field lacks these large, rich data sets that include the clinical information and treatment and genomic data that can be useful to answer research questions. We can think a lot about ways AI could be useful if there were these large, high quality data sets to learn from. For example, AI could help improve diagnosis and understand pathology. It could possibly predict treatment response based on patterns of response in existing data. And in the same vein, AI could suggest a specific therapeutic approach based on what data has shown to be effective. We could also use AI to screen drug libraries and search medical data, learning from these complex data sets. But we can only get out of AI what we put into it. This means that we need more high quality, structured, real world data sets from sources like the registry and other registries and observational studies. Patients can contribute to this growing source of data by participating, joining, and sharing their data in ways that the AI can learn from. If you are someone who's intrigued by these kinds of platforms, for example, like ChatGPT, and how you could use it to understand your own health information, we would encourage you to instead consider an opportunity like XCures that Denise mentioned earlier. This is an AI-assisted healthcare data platform that's designed to extract and structure your clinical data from medical records to help you and everyone else affected by EHE in the future. In this way, your data contributes to the larger goal of realizing AI in healthcare. And I'll give you a minute to scan this QR code now. If you're interested in the future of AI in healthcare and research, this is a really great opportunity to safely and securely contribute your data to this area of knowledge. AI is just one of many avenues of research that are really ripe for exploration right now. We've made so much progress in the last several years, and that progress has identified even new opportunities to learn more. Immunotherapy approaches are making significant advances in other cancers and are just now beginning to be studied in EHE, including that PD-L1 inhibitor study in Canada that Denise mentioned, and actually some new research we've newly funded out of Germany. You may recall an educational session we hosted late last year also on histotrypsy, which is a technique used in other liver cancers, but has not been specifically studied for hepatic EHE. We have an opportunity to study exactly when and with whom this technique should be considered. Also for liver EHE, it's not yet definitively understood which patients get the most benefit and when from liver transplants. This includes those with metastatic and with local disease. And a few times throughout the conversation today, we've discussed forms of biomarkers. While there have been some exciting early findings, there's currently no clinically available, standardized, and well-studied markers that inform who's benefiting from therapy or when disease might become aggressive. This is a great need that holds significant promise to improve treatment approaches. And as an organization, this is an opportunity we are especially interested in actively exploring in research areas. And speaking of biomarkers, blood-based disease monitoring like ctDNA or cfDNA assays are a growing area of interest in cancer biology across the spectrum. And while we've learned how other tumors can be detected using these tools, research hasn't quite yet shown that EHE can be detected in this way. All of this means we need more studies, more data, and together they can help us to achieve the better outcomes that we strive for.
Okay, we are nearing the end of our presentation, so thanks for your patience and walking through this with us. One of the things that I want to say here, for those of you who know me, you know I'm passionate about research, but we at the foundation, we are passionate about amplifying your voice and your experience. This is what we're here to do. it is important to us that we represent your needs and your priorities. People affected by EHE deserve to have this, and we need this. We need to be partners in making sure that the voices of people with EHE are amplified everywhere, primarily in research every day. We work, a lot of our effort is in encouraging and facilitating research collaboration. It's an important part of what we do. It's not something that people can see that we do every day. It's not something that you can see that advocates all over the world. Fiona has spoken up a couple of times. She is such a great advocate in Canada for research and in partnership. And you know those all over the world that are, and everybody's working hard on this. Our job is to provide supportive and accessible information. So everything that gets published, everything that we find about, we want to share that with you through our library, through educational sessions, through experiences. So that's a job that we have to do, and we are so glad that we have supportive people to do this with, and patients who are interested in research, in not only being part of it, but in learning more about it. Engaging drug developers and regulatory agencies, I've talked about this already. I mean, we do that every day, all day. Our foot is on the gas to make sure that drug developers know who we are. They know who this community is. They know that we are a research-ready, engaged community. That makes a huge difference when you're talking with drug developers to know that they can study a rare disease. One of the things that we do, this is not the fun part, but representing voices in policy dialogue. Policy is frustrating for everyone, but it is a need. And so by harnessing patients' experiences, understanding the burden of the disease, understanding needs, that gives us what we need to inform policy dialogue. So very, very important. And really, you know, one of the things that we do is, and people always know, I want you to know that you can contact us. Helping people who are affected by this disease is important. If you are in your journey and you don't know where to go, you need a new care plan, you need a new care provider. If you're newly diagnosed, it doesn't matter where you are, you can contact us, and we're here to help and answer questions as best we can. Our contact information is here. Scan the code. You can find us all day, every day here, and we'll be glad to put you in touch with, if it's not our foundation, we'll put you in touch with people at other foundations around the world to get you the best help that we can. I want to just give a shout out to our partners in advocacy. I mentioned our folks, the EHE Rare Cancer Charity, the EHE Italia, the Australian Foundation, our Canadian partners, and importantly, a congratulations to the people in Germany for a newly announced organization to support people with EHE in Germany. And we are so glad to be partners in our mission with these people. There are other icons here, logos from other organizations. This is just, again, it's a scratch at the surface because we don't have all of our academic research and clinical care partners on here, and they are so important to us. We can't do the work that we do without all of these people, and we really can't do the work that we do without you. So thank you very, very much. Kudos to all of you for being here today on a Saturday. We hope that you're warm and snuggly, you have lots of coffee, and you've enjoyed this conversation. And now I want to turn it over to Q&A. I'm going to, if Christy and Maggie agree, I'm going to stop sharing my screen because this will make it easier for us to do Q&A. I can see some people. I see Dr. Lamar. Thank you. Thanks for coming, Dr. Lamar. Good to see you. That's great. I see a lot of names of people. I'm going to jump over. Come off. If you want to turn on your cameras, I welcome you to. If you don't, that's fine as well. I want to go to some of the questions that were submitted as part of, during registration, people had the opportunity to submit questions, and I want to go to some of those. Let me see. I'm going to get Maggie and Christy if they need to, or Christy, you can help me with the chat too. I'm going to
Mhm.
pop that open because I have too many screens open here.
Yeah, there's just a few questions in the chat, not too many.
Yep.
They're more towards the top, Denise. There's just a few up there.
Got it. Yeah. So I'm going to jump through these Q&As that came in. So there was a question about which treatment option is currently proven to be more efficient when EHE progresses. Someone says that their oncologist said that studies show efficacy, for example, in sirolimus. And yes, so we pointed to that data when we were looking at that earlier. That is as much as we know. What we've shared with you is as much as we know, and it is as much as doctors know at this point in the game. So I would say, you know, the data that we have, the consensus paper on EHE does point to sirolimus as the most active agent systemically for EHE. And I would say that, you know, in your treatment journey, where you are does matter. So context matters. Some people will have had a treatment and maybe they couldn't tolerate it or failed it, and then they have to go to another treatment. So have those conversations. Someone asked if a person's diagnosed through in a body part through a biopsy and then have small pulmonary nodules that appear compatible with the disease, is a lung biopsy required to confirm that it's EHE, I'm assuming, in the lung. So I can't give you that as a medical advice. I can tell you that what we hear from clinicians and what we hear from people who have been through this journey is that you know, elective biopsies in EHE, and I would consider this to be somewhat elective. You can assume that if you see something that it probably is EHE. That's what most clinicians that we talk to would say. But that is, again, every question has context that we can't answer, and your doctor will have to make that decision. We would not suggest biopsying everything that is found in someone, because sometimes there are unintended consequences to biopsies that can't have been planned for. And then what do you do about that? So we would say act like it's EHE. Someone asked if you can be eligible for a clinical trial if you've had a liver transplant. So that depends. Some studies will allow people with liver transplantation and some studies will not. That is a case by case. So if you're considering a study, you can look at the criteria. Sometimes it will say, sometimes on those clinicaltrials.gov sites, it's not as clear. Contact me if you want to learn more about that. Christy and I can help you answer that question. But it is usually very clearly specified in the clinical trial protocol if a transplant can be included or cannot be included.
That's also a great conversation to have with your physicians as you're going through your care process, is talk about what the consequences are of the treatments you're choosing now. Will the treatment that I'm choosing now, or lack of treatment that I'm having now, will that affect my ability to have a treatment later? That's a great way to have a conversation with your entire care team about what your next steps are going to be.
Good point. Good point. We have a question about diet. So keto, fasting, and other supplements that could be used to help treat EHE or help control the disease. And you know, there's no data on this. I mean, this is a challenge across so many cancers. It's just knowing, you know, what could you do other than having chemo or having another intervention that could help control your EHE. And there's just no good data out there on that. I think that this remains a highly personal decision. And certainly if you are making dietary changes, significant dietary changes, particularly if you're taking any medication, I strongly encourage you to be transparent with your doctor about what those are. Make sure that all of your doctors know what drugs you're on, and if you're changing your diet or you're doing something. Things like green tea. Green tea has effect on some drugs. Grapefruit juice. it's something that some people enjoy, some people detest, but it does have an effect on drugs, on the metabolism of drugs. So your diet can affect your care plan if you're on a therapy. So make sure that you're talking with your doctor about that. There is a question about personalized vaccine being offered to patients for cancer. We don't have any knowledge that that has been investigated in EHE, reported in EHE. So I would say that that is a futuristic question that should be asked. Immunotherapy is something that is just starting to get studied in EHE. So we really want to make sure that we learn more about how these types of treatments could be important. We've actually we've just engaged in some discussion about EHE, translational research, and we look forward to learning more about that. So stay tuned on that one. Let's see. There's a question about moving from active surveillance to treatment. The question is, when do you suggest moving from active surveillance to treatment? Again, we could ask 10 doctors and they'll say it depends. But I will have to say, overwhelmingly, the consistent response is watch symptoms. So you can look at how your disease shows up on your scans. That is one way. That's the classic way that doctors want to see progression of disease or stability of disease. And that's a great way. But any change in symptoms in EHE specifically should be noted and talked with your doctor, because that could also indicate progression. It could be something else as well. But we do know that pain is a widespread symptom of EHE, and that pain may or may not be localized. So it's hard to describe and should be assessed probably by a pain management specialist to understand what the source of the pain is. But there are other symptoms of EHE, shortness of breath, fatigue, and in saying that, you all know that these are symptoms of other diseases, but every symptom should be investigated, and you might use a coordinated approach to see, is my disease changing enough that I need to consider therapy or not, and have that conversation with your doctor. I wanna point out here, because there's a question in here that really speaks to what we talked to today. Someone's commenting that they have been to multiple top EHE centers, and they got conflicting answers on how they should treat their EHE. And then how does this discrepancy pan out? And this is really why research is so important. That's why observational studies are important. It's why preclinical work that's being done is really important to support and help read out what treatments could be better than others. The data, the observational data, understanding what happens in a very defined set of patients prospectively or in observational studies over time is really, really important. The data table that I showed that had all those different data on it, this is why you get, I always say, if you ask, 4 people, you'll get 5 answers, because it matters. But every single patient experience changes that game. I'll invite you to raise your hands, and Christy or Maggie, do you want to read out the questions in the chat? I apologize, I lost my chat somewhere down here.
That's okay. I actually just saw a great question that I think is also related to what you were just talking about in the chat. That sometimes doctors don't understand, or better yet, can't really explain the pain that is a common symptom for so many folks with EHE. And first, we'll just say you're absolutely right. That is such a huge struggle. It's a pain, understanding pain as part of this disease process is really, really complicated. It's also one of the things that EHE Foundation as a whole is looking into as part of the registry. We're trying to collect that information to make it easier for the clinical and research community to understand how the pain that so many people are experiencing is contributing to the progress of the disease. So all that to say, you're definitely not alone in this experience. I wish we had better answers, but we're trying to collect them to understand it better.
Robin has a question in the chat about the connection between sirolimus effectiveness and the location of EHE. I don't think that there is strong data yet regarding location involvement, but we do know that for patients who have pleural involvement of their disease, particularly serosal effusions, those patients have not shown to benefit significantly from sirolimus. So that has been, that's probably the earliest finding that we have. But we know in the data that there are patients who have lung, liver, bone, they have metastatic disease, and different progressions of their disease. And sirolimus has been showing early data, small data, to be effective. So I don't think that there's any way yet to point to a location of disease.
Denise, can you talk a little bit about some of, you kind of introduced this a little bit, but can the contacts that we have and the interactions we have with folks in other areas of the world, especially in Europe?
Regarding drug development?
A little bit of all of it. I think that would be important to talk about. But this question in particular is talking about the European specialty centers and our interactions with the European specialty care centers.
Yeah, so there's a good directory on there's a website, it's called SPAGN SPAN, and so they refer to reference centers. So these are sarcoma specialty centers, and truly I can't highlight enough how important it is if you are diagnosed with EHE, if you or your loved one have EHE, where you're treated matters. And you can get treatment. I like to call it care sharing. You can have care at your local hospital shared with an expert. It's really important to have informed care and having a sarcoma reference center. So again, Europe has this nice network, and these people have kind of been qualified by some, I think it's self-report, that they are expertise in sarcoma. And they have, they will have surgeons, they'll have radiation oncologists, they'll have pathology, they'll have medical oncology that all have focus in EHE. So if you're, no matter what country you're in, because there are experts in many countries, there are countries where there is not that expertise, but I encourage you to please seek out that care and at least consult with an expert on some regular basis. It might be once a year for you, it might be every 6 months, it might be every 3 months. I realize in saying that there's a high burden to get places. That's also why we work with clinical centers to try to, we send the consensus paper, we talk with people, we try to develop more oncologists who can work in sarcoma and work in EHE by talking with them and inviting them to learn. So that's really, really important. But yeah, you can check out the directory, the country directory, and you can get to that also through our website. I want to ask, so I'm going to put Dr. Lamar on the spot for a second. There's a there's a question here about statins, and John, we referred to your work lightly in this research. I don't know if you want to say a word about that at this point and just answer the question. It just talks about what is the research and what other drugs can be combined with them.
Sure, thank you. So, our research has not been published yet. I should start by saying that. So, it's always important, I think, patients to kind of just pay attention to where the source of the data is coming from. And so we as scientists do our research, and then we have other scientists evaluate it and make sure that we're doing everything right and that we've interpreted it right. And so before it gets published, it's only been vetted by us and some other people. So so far, what our research has shown is that that statins can benefit mice that have EHE. So we've given the human EHE cells to mice we've used the cells from the Stacchiati group that you saw in that slide with the sirolimus study. And so when we treat those mice with those tumors with statins, we see that the tumors grow quite a bit slower than they do when they're not treated with anything. And Sarah is on the call. She's the student that's been doing all this work. I just get to look at the data that she brings me. So good job, Sarah. Sarah spent a lot of time trying to figure out kind of how that's working, and she's got some good data on that as well. So hopefully we'll be able to get that paper submitted and published. I think there are still a lot of questions that Christy mentioned on that slide that we don't know. I think the one thing we are missing is clear evidence in patients that this is potentially working, and this is another area where the registry could be helpful to us. So, a lot of patients just happen to be on statins to control their cholesterol or for other reasons, and so if you happen to be on a statin and that data is being collected by the registry, that may sort of help us try to understand how different patients are responding. I think it remains to be seen whether we can set up some sort of trial to see what will happen to if we put patients on statins. But I think the first step is to kind of answer some of the remaining questions that Christy brought up. There was a retrospective study on EHE patients that was published as an abstract, so that's not really peer reviewed either. That did suggest that patients did benefit a little bit better, or patients who were on statins had slightly better survival than ones that weren't. But again, that study kind of suffered from the same problems that Denise and Christy have mentioned, which is that there weren't enough patients on the study. They weren't really all the data wasn't really all collected in the same way, and so it's kind of hard to really make any conclusions about what that meant. But it shows maybe there's some promise. And then to answer the second part of that question, you have to be very careful about combining any drugs together. You should always ask your care team about that. We're looking to see whether statins can be combined with other studies, and Sarah's done some really good work there too. So we're looking to see what it does in combination with the TEAD inhibitors, for example. But sirolimus and statins cannot be taken together. So some drugs have toxicities that get amplified if you take them together. So you have to be very careful. You have to check with your care team. Don't go out and put yourself on something like a statin, which is pretty easy to get access to without checking with your doctors.
John, thanks. I'm glad you mentioned that. I was going to say the same thing. That's really important to note, and that's why we've just working with clinicians, we know that patients underreport what they're taking, and whether it's forgetfulness or you just want to see if you can, take one more thing, but there can be an adverse, part of that. So make sure that you talk about it. But statins and sirolimus cannot be combined. And I believe that if you're taking sirolimus, Dr. Stacchiati highlighted in her talk with us last year that green tea is not your friend. Green tea can be your friend in a lot of other situations, but probably not your best friend. As it turns out, diet does apparently affect sirolimus quite a bit. So something to just keep in mind. We'd like to learn more about that. I don't know if there are wide scale studies on that, but really good. Are there any other questions in here? Please, if you want to come off camera, if you have a question and you want to raise your hand, we welcome you to ask your question live.
Denise, I just want to take 2 seconds real quick. There was a question that came through about the cause of EHE. And I kind of mentioned this a little bit towards the very beginning, but I just want to take a minute to say it explicitly. There is no hereditary origin of EHE. This is not something that you're going to pass on to your children or you got from your parents. And the only thing that we know of that causes EHE is the fusion that happens completely spontaneously. So we don't know of any other cause. There's nothing that you did that brought this upon yourself or anybody else did that brought it upon themselves, and you can't pass it to anyone else. So just want to make sure that that's said out loud.
And that's an area, thanks, Christy. That's an area that we're in the process of updating and adding more questions into the registry, just to ask questions about people's lives more. We didn't add that early on. We don't have any scientific reason for adding it, but registries often ask you about, your occupation, where you work, et cetera, just to understand if there are any environmental effects on any diseases. So we're adding more things into the registry.
Denise, I had a question.
Yes.
It was about the assumptions we make about sirolimus or all whatever way we pronounce it. Can we make equal assumptions about its sister drug everolimus or what is
Yeah.
it? Ifinator?
Yeah, that drug has not been well studied in EHE, Fiona, and
Okay.
And what I can say is that I know that I've talked with people who have been on sirolimus and they had bad side effects, and their doctor changed them to everolimus, and they didn't have the same side effects. So there's something different there, right? Just to have that happen. Like sirolimus, everolimus is also not readily available to patients. So we still have the patient access issue. Sirolimus has been just kind of the easier to get, and I think more widely prescribed. There's another there's a third agent in this same lane, and the name starts with a T, and it escapes me right this second. But no, there hasn't been a lot of data on everolimus. Things like our observational studies should read that out. So because we know that there are a lot of people on these drugs, but I think that we would see in our observational data, I suspect we'll see that that is more prescribed for people who have had a liver transplant than people getting that as like sirolimus who've not had a liver transplant. So we have mixed data right now in our registry. We can separate that, but we know we have people who have had a transplant that are on these drugs, and then we have people that didn't have a transplant that are on these drugs. Because keep in mind, that's their primary use is for organ rejection. We'll see what we learn through those data.
One of the ways you can think about it too is kind of like how we were talking about the TEAD inhibitors and other classes of drugs where there's lots of different drugs that sort of work the same way, but they are slightly different from each other. The same thing is true with sirolimus and everolimus. They have a similar goal in the way they work on certain cell signaling pathways, but that doesn't mean that the way they get to that goal is exactly the same, which is why some people respond better to some than others. And we're actively learning about all of these big classes of drugs that are similar but still slightly different from each other.
Thank you.
Mm-hmm.
Yeah, I do want to come back to just the clinical trials questions, because there were a lot, and a lot of them are the same. You know, at the end of the day, people want to know more about clinical trials. They're interested in knowing if they could benefit from a clinical trial or if they could get on a clinical trial. The information about each of the clinical trials can be found online, but if you really are considering looking at a clinical trial as part of your care plan, I encourage you to talk to your doctor first, to just see, because they can help you see if there are any blaring criteria that you might not meet. And some of the criteria have to be medically assessed. They can't be assessed by you or me or anyone without touching the patient and knowing some of the lab values of a patient. But then, you know, the next step, if your doctor and you or whomever you trust, it doesn't have to be your doctor, but if you talk to somebody and you say, I want to learn more, get in touch with us or get in touch with a study team if their information is online, because it is really a one-to-one discussion. Each person, particularly in this ultra-rare disease, is a unique consideration, even though there are very clear protocol criteria. And think about that as you consider that, because as you know, we say consider this, but consider the travel. Consider that there is a requirement when you join a clinical trial. There's a large requirement on your side to be there and to undergo those assessments. You do get very careful care under a clinical trial because it's highly controlled. But just keep that in mind, and we're glad to talk with you about any of it.
Denise, someone did actually message me and ask, is the NAB sirolimus trial at Dana-Farber actively enrolling already?
It is.
now?
Yeah.
Okay.
So they just, that site was just activated last week. I got word that it was active. That's not like, I don't think you can publicly find that anywhere. So I'm just telling you that today.
Hot off the press information.
Hot off the press. And Dr. Michael Wagner is the lead PI there. But if you are under the care of any oncologist there, you can ask anyone there. They all know about it. We've all been on calls about this. And then it's going to open up at other centers in the United States. It takes a while. We have a lot of paperwork to go through to get each site opened up. There will be, you know, a limited number of slots early on. I'm not really worried about anybody getting turned away at this point. I think that, you know, if people are interested in this, but I can talk with you a little bit more. And if you're in the Northeast anywhere, if you're from New York over, And if you live in Pennsylvania, you could easily get here too by train. But if you want to come to Dana-Farber, we're planning to have a little education day there on April 11th. It'll be a couple of hours. We do want to give Dr. Wagner a chance to talk about this clinical trial. But if you miss that, don't worry. We're going to keep bringing him to have him come back and talk online, virtually with the community so that we can learn more about this study, and people can decide if they want to participate in it.
Denise, I'm going to start playing you off because we're at time.
Okay. Thank you.
Yeah, go ahead.
No, I was just going to say, we really appreciate you all coming here today. I can't say thank you enough. Thank you to Dr. Lamar for letting us put you on the spot for just a second. He is, he's one of our back in the lab heroes, and Sarah, who is with him, who also, Sarah has given a lot of time to EHE, and we greatly appreciate everything that they do in their lab. Fiona, thank you for shouting out several times. We're going to add you to our mix next time so that you can do part of these presentations. She's good at it.
I'm so sorry. I hope you didn't mind.
Nope, you're great.
Not at
You're
all.
always great. Thank you so much. And we just have to give a huge thank you to our partners, again, advocacy partners all over the world, who help make all of this possible. Scan, call us. Most of you probably have our email and phone numbers, and we encourage you to call with any questions and follow up. If any of this sparked new questions, send us a note over the weekend.
And
thank you so much.
This has been recorded. It will take me probably realistically 2 weeks to get it captioned and put back out on our website, YouTube, social media channels. So just watch our news and our social media, and I'll put that announcement there when it's ready.
Yeah.
And for me, it was such a pleasure to meet all of you and see your beautiful faces. And I look forward to interacting with you all more over the next weeks and months to come.
We are so glad to have Christy. Thanks so much. Thank you, everybody. Thanks for giving us part of your Saturday. We really appreciate it. Be well. Take care.
Thanks, everyone.
Bye-bye.